So, um, Good afternoon, everyone. Welcome to the H3D Foundation webinar series. Our today's speaker is Dr. Godwin Dziwornu. Dr. Dziwornu represents Holistic Drug Development and Discovery Center, H3D. H3D operates within UCT's world-class translational medicine research environment. Dr. Dziwornu has expertise in organic chemistry, medicinal chemistry, and natural products, and is involved in designing novel chemical series for exploration as anti-infective agents towards a drug discovery venture. Before we start with the talk, I would like to let you all know that we will have a Q&A session after the talk. So questions can be posted in the chat or kept for the end. Today's topic is From Nature to the Lab to the Clinic. What is the current state of art and science? And now I would like to request Dr. Dziwornu to enlighten us with their work. Dr. Dziwornu, over to you.
Thank you so much. Thank you, Sweetie, for the invitation in the first place, and then to the H3D Foundation team for the opportunity to present. I'm also grateful for the kind words and the kind introduction. So yes, today I want to bring a subject or remind us of a subject that we are very much aware of. Once in a while, I'm sure we've thought of it as medicinal chemists or as scientists. We've had this thought somehow, being humans and loving nature. We've asked ourselves questions about herbal medicine or all of those kind of questions. Even the tea we drink, whether it is rooibos, whatever it is. I mean, things of nature and the clinic and our health is very important to us. So I can imagine that this title was received with a lot of eagerness to want to hear what I have to say, and I'll just go straight into that. So when we talk of a natural product, so generally it's any substance that you can obtain from nature. So it's a product from nature, and so you say it's a natural product. Okay, it can refer to any biological part of an organism. It could be extracts, it could be formulations, it could be isolated chemical compounds. So the leaf of a plant is a natural product in general terms. When you make your rooibos tea, one will consider that extraction or what you have, the tea you have or the coffee you have, as natural product. But usually the narrow definition has been where in the research, in chemistry research, we tend to consider it with respect to isolated chemical compounds. Okay, but generally it is any product from nature is a natural product. So in its more narrow definition, one will consider primary metabolites and secondary metabolites. So primary metabolites is in everyone. I mean, every part of nature has it. So every living part of nature. So carbohydrates, fatty acids, amino acids, these are the very ingredients of life. Okay, so our life depends on that. Photosynthesis from carbon dioxide and water making glucose. It's a primary metabolism. Okay, so then the glucose is a carbohydrate. So that is something that is in every plant, even down to the sea, everyone makes it. What is different is the aspect of secondary metabolite, which tends to be specific with species. Okay, and that would depend on in fact, that's what defines a species. So that is what will make a flower a yellow flower, and a plant produce yellow flowers, and then another plant produce purple flowers. And so you can distinguish between two different species. It's with respect to the secondary metabolites they are making, which goes down to their genes or the DNA that they are composed with. And usually when it comes to research-wise, we are more interested in the research or drug discovery. We are more interested in the secondary metabolites that are produced by these species. So basically, this is just for our information. Primary metabolites are what organisms make use of to make secondary metabolites. So that in some compounds, in some secondary metabolites, you can clearly see a glucose or a sugar molecule in there, for example. Okay, I'll show an example in the next slide. So this is just to give us an idea of what kind of secondary metabolites are coming from what kind of process or primary metabolism, the breakdown of these primary metabolites. Okay, now these are examples of secondary metabolites, what one will call natural products. So these compounds here, these A, P, and X, dicapitalin A, P, and X, is in fact from the dicapitalum species. It's a plant species that is in Ghana or in the tropics. Just recently, researchers in China have isolated very interesting compounds in this related to these compounds. In fact, South Africa is home to this anti-cancer drug, which grows in the Eastern Cape. This compound here is responsible for the bitter taste of the melon that you would find in the Namib Desert, for example. This compound here was isolated from what is called the Okna plant, which is in Mauritius, in fact. All these other ones came from seaweeds from Port Alfred in the Eastern Cape, but from the Marion Islands, which is part of South Africa, but then is close to Antarctica. Now, these are compounds that I have worked with or I isolated as part of my master's or my PhD. So they are just a summary of what you can find. But then the big question that I want to bring, which is in fact the focus of this talk, is of what value are these? I mean, why go to Antarctica? Why go to far away to the forest, deep in the forest, to get these molecules and then just isolate and publish them? Because for now, they are just on my laptop as published compounds. Of what value is there in the pain? And this is where I want to answer the question to say the value of an isolated secondary metabolite, okay, is in what they can become if time, energy, resources are prudently, and prudently, I would want to underline that, invested in them. Okay, so it's just like gold. I mean, extract gold or mine gold, it's of no value until time, energy, resources are prudently invested in them. And the same to do with people. Now, that is what I want to focus my attention on in this talk. And so the title, From Nature to the Lab to the Clinic. Okay, now just to bring us back to medicinal chemistry, I don't want to do natural products research or chemistry today. That can be for another webinar. But hit identification in drug discovery takes basically these four approaches. And I'm borrowing this slide from Jessica, who delivered a good webinar some time ago. I found it very useful. So with natural products, yes, as we've said, they are sourced from plants, microbes, marine organisms, and toxins. Of course, you go through isolation, characterization, and all of that, and then you can do synthesis, semi-synthesis, and of course, these are processes, but I don't think the medicinal chemists need to go all the way down. You do not have to go all the way down to isolating and characterizing, which could be challenging, I agree. I think that, in my opinion, there are more than enough small natural product molecules that one can work with. But then the two big questions are, what have you found? Have you searched? Okay, so in what we do as medicinal chemists, or drug discovery researchers, we are looking for a new chemical matter. Okay, what I found is that usually medicinal chemists would want to stay away from natural products because of complexities like structural elucidation, but we do not have to start from there, is what I want to emphasize. What have you found? Okay, just a Google search to say anti-malarial indole compounds, you'll be amazed with what data or literature would pop up. And you can prudently go through and select a molecule you can use as a starting point. But it is in what can you do with what you have. Okay, what makes natural products bioactive? I think that this is a question that lingers in our minds for most people. And I just want to pick 3 points to explain that. Okay, So these are made by proteins or enzymes in the body. Okay, now because they are made by enzymes in the body, they are made such that they fit the enzymes active site. Okay, so they are complementary to they are like the lock and key. Okay, so take the protein as the lock, and then the natural products that are made as the key. They are well fitted. Okay, so that is how they are produced. But the thing with it, with that kind of a system, is that nature or organisms tend to share species in the same genus, for example, will tend to share similar proteins across board. Now, that is even just in the genus. Apart from that, you can have several organisms share similar proteins, which means that a natural product produced by one enzyme could fit, okay, partially or fully with the enzyme in another organism, for example, because of how nature is intertwined. And that is why you can have a compound from plants being active in humans. Okay, so it comes from the fact that it's the lock and key sort of idea between the protein that makes the enzyme, the natural products, and the natural products themselves. Now they are three-dimensional in form because the active sites themselves are three-dimensional. Now this gives them access to membranes in the cell, and so they can really permeability tends not to be a big issue to natural products. And then I think this third point is also very important. Organisms will make compounds to defend themselves or keep them in continual existence, which means that take the sea, for example. The water is huge, but it's out of the sea that we found some of the very potent anti-cancer drugs, because the small fish or organism there or bacteria there needs to make that small molecule that even when it's diluted by the big sea can still be potent. If you understand if that makes sense. So yes, the organism makes that because it wants to defend itself and it wants to keep on its evolutionary existence. So that's why we would find some three examples that we will find natural products to be bioactive. Now let's come home and let's begin real drug discovery discussions. Okay, so annually or over time, there are these natural products chemists who report on how successful natural products are in the drug discovery with respect to drugs that have been made or that have been approved. Now you will find that between 1981 and 2019, you have 36, almost 37% of purely or natural product related compounds. And I will explain that in this chart. And then purely synthetic drugs. Synthetic drug means you would add to the natural product. Now, what are natural product related compounds? I would want to emphasize on these four. So N here refers to purely natural products. So take something like artemisinin, purely natural product. You do nothing to it. It moved straight from nature and was able to get to you didn't do anything, it got to the clinic. But see the percentage of that over the period, just 3.8%, telling you that it's less likely to have that. You are not likely to. I mean, it could be only by serendipity. Now, natural product, ND refers to natural product derived. Now, this is where you derive derived from a natural product, and then you do modification, you modify it. So you take it, you realize it doesn't have certain qualities. Perhaps the PK properties are bad, it's not very soluble. So you bring it into the lab, you modify it, and then improve on its properties. You can take dihydroartemisinin as an example. So artemisinin itself is the pure natural product, dihydroartemisinin, or some other modifications. I mean, you can find a few of them. The next one you would find is synthetic, and then there are mimics. Okay, so you can have so this is where you would have things like your tea, but these are the traditional Chinese medicine, where now you can have the leaves themselves, but they've gone through toxicology studies, and so they are given. And then one can take it as approved drugs. The percentage is low because that approval just started recently. Now, this S star refers to a natural product you took the pharmacophore. Pharmacophore meaning the part of the natural, not everything in the natural product will be potent, but you select the most important aspect of the natural product and then you modify it. Now, it's all four of these put together that gives you 37%. But the emphasis here, the point I want to make here is, if you want to move from straight nature to clinic, you may just have over this period, 4%. But the modification and doing something to it, or adding, gaining some knowledge from the natural product, rather gives you more. It gives you about 33%, so to speak. And hence emphasizes the need for the lab process. Okay, just to show this again, so antibacterial drugs by source. Here you will find that again with natural products here, how many natural products within the period have been approved as antibacterial drugs? Just a few. But then with derived, you see that there are more. Okay, so just in 11 purely natural products, but 78 of ones that have been modified. In antifungal drugs, you do not even see natural products at all. Okay, here you rather see ones that you do a mimic. You pick the pharmacophore and then you develop it. And when it comes to antiviral, a similar case, you don't find purely natural product. So what this tends to inform us on is, for example, if I want to venture into antifungal drug discovery based on natural products, I would be cautious, because then this data suggests to me perhaps a parasite or fungi are not really susceptible to purely natural products. That doesn't mean you can't find any, but the journey to the clinic, you may not get there. Okay, so just to put this in context. Now, what makes a good hit? Let's say you found a natural product, you have isolated it, or you found one in the literature. What should you look out for? How do you start? Again, this, I would like to acknowledge Jessica. I took this from one of her slides. So you would want to look at the potency and selectivity. Okay, I will not go into details with this because as medicinal chemists, I would assume we know much of this. But you would want to see the ligand efficiency, how specific is your compound to the target. You don't want it to be promiscuous, hitting everything. No, that is not good if you are taking it into humans. Now, what is the drug-like properties? We can talk of Lipinski's rule. And here I just want to remind us that when it comes to natural products, natural products do not really fit into Lipinski's rule, because nature does not respect just lab compounds. I mean, data or conclusions made from the nature does its own thing based on what it needs it for. And then synthetic accessibility and versatility. How easy is it to synthesize? So when you want to choose a natural product to start a project, okay, you would want to consider all of these. Okay, then there is what we call toxicophores. Are there fractions of fragments or moieties in the natural product that could suggest toxicity issues? They could be reactive metabolites. They could just hit everything. Again, they are promiscuous. They hit every enzyme. Now, if you have this as a benchmark, okay, and then you find compounds, you are able to, in fact, triage right from the beginning. You're able to pick up what you should pick to continue your work that you want to do, medicinal chemistry work or drug discovery work, or not. So this I find very, very, very useful. Okay, so I want to ask a few questions here. So what next, based on just what I have said, I mean, in the last few slides, what do you have in your hands? What have you found in the literature? What compound is it? What is the size of the molecule? Does it have hit qualities? Okay, now these questions are important because you would have to start from somewhere. Your chances of success will have to go through a lab, okay? You need to add value to what you have. If you run a campaign with only synthetic compounds, well, that is just half of what you can. If you can have natural products as part of your starting point, it is also viable. It's something that you can use. Now, what approach do you want to explore? Do you want to make it a mimic? Do you want to make it by a derivative? Do you want to do semi-synthetic modification, or you want to pick the pharmacophore? Now you look at the molecule, you look at what you have in your hands, and then decide right from the beginning how you want to approach it. What will your lab research look like? The third question is, what resources do you have? And this is where I get passionate about it, about this topic, and why I want to present it here. Because in Africa, the default for us is natural products. It's easy to order bulk solvents, and so you extract from plants, and then you get crude extracts, and then you publish, and then that is all. We get a lot of things. So this compound has anti-malaria, it has blah blah blah, but you don't add any value, because what resources do you have, and where can you find help? And I think that the core focus of this talk is on point 3 and then point 4, because I believe I would have convinced you with point 1 and 2 in the previous slides. So let's look at points 3 and 4 more critically, if so. So what does present day natural product based drug discovery look like? Okay, as I did say, I mean, some years back, maybe 30 whatever years back, when people were enthused about the beauty of these compounds and how different they look, it was just isolate, publish, isolate, publish. But now we've isolated, we've published, we've seen almost all. So there isn't so much excitement anymore. Okay, the real concern or science happening now is value, adding value. But if you want to add value, what does it look like? Okay, I found this image online which I felt represents everything. Okay, it captures everything in it. And just by it, I would assume that it just hits you as much as it hits me. A lot of stuff needs to go into it. A lot of detail needs to go into it. Okay, whether it is isolating it or you're getting it from a library, you're screening to get what target, mode of action studies, the synthesis, the medicinal chemistry synthesis of it. And then before you even do in vivo efficacy and think about developing it further. So this again is what goes into it. So you can think of current day natural product based research as being what was done with drug discovery a few years ago. So if I should say natural product has caught up with present day drug discovery, and there is no room or space to overturn it. I mean, this is what you need. Now the question is, do you have this infrastructure, or you just want to just go there? We need, as Africans, we have infectious diseases, we need to solve them. But I think that I have good news as to how, as Africans, or whether there amongst the audience there is anyone involved in this, how can we get to overcome this? What resources are available on the continent to help us in this? Okay, so the next step after what I have shown in the previous slide is to know that natural products research is entering the 4th industrial revolution. What that means is that now if it is happening now, plant collection, you will not need to take a botanist into the forest anymore. Let me put it that way, to identify your plant for you, because now there are apps that you just show. I mean, you just take a picture of the plant and then suggest to you what the name, the scientific name, or what the plant is. Okay, so we have to move at the pace of scientific research. I mean, or the pace of technology, if I should put it that way. So we cannot be doing the same old thing. We need to catch up. Okay, now this is where I want to deal with a case study. As I did say previously, we have the resources in Africa to do this kind of work. Okay, now H3D is one place that, for all that big image I just showed you in the 2 slides back, you will find that we have these resources here that can support this kind of research. And that, in fact, is the main core of what I want to put across. Okay, now I want to deal with a case study where as H3D, or in fact, from the academic group of Prof. Kelly, this natural product fusidic acid was worked on. Okay, now it was worked on going through the cascade, as I have shown previously. Now, fusidic acid is a natural product. It was isolated from a fungus. It's been used in the clinic since 1962. It is that natural product that did not need any modification. So right from nature ended up in the clinic. A lot of lab work was done, but no, is this the perfection of nature? No analog made in the lab could meet the properties of fusidic acid. And its antibacterial activity are just well published and well established. Its mode of action as inhibiting elongation factor G in bacteria that prevents protein synthesis has also been documented. Okay, now there is no cross resistance with some of the known anti-TB drugs that are on the market. I mean, clinical isolates. So the question is, what did we do with fusidic acid? Okay, Prof. Kelly, led by Prof. Kelly, of course, who is the team head, brought this compound into the lab with the idea that based on the properties of the company, as shown in the previous slide, one could do medicinal chemistry work. Okay, now this is where you look at the molecule, as I did say, what do you have in your hands? This is fusidic acid in my hands. What can I do with it? If you want to do the total synthesis, you may not have time. Medicinal chemistry, you may not have time for that. But can you take advantage of functional groups in the molecule and then move your compound for it, make it add some more value to it? Now, here you will find that fusidic acid had what someone would say not great malaria activity to start with. But just by making simple modification, you can drop the activity quite low, okay, to something that is encouraging. And the Prof. Kelly's group, with resources and the support from the facilities, the infrastructure built in H3D, could do running of assays, okay, testing these compounds, whether it is all of that, I'll show in the next slide. So the chemists did a lot of good modifications. Now, this is talking about part of what we have. So the computational facilities that we have, so that by 3D quantitative SAR modeling, the team was able to suggest compounds that could be active, making use of 61 compounds. So they predicted these compounds, came to the lab, made them, and the compounds were in fact active. And you can see the potency as represented here. In fact, in docking studies, the team was able to rationalize why this analog compound 2 was more potent than fusidic acid itself. And here too, you can see that it picks with this oxadiazole, is able to pick several more interactions compared to fusidic acid itself. Simple modification, but then adding value to what you have. Okay, that is with antiplasmodium or in the malaria space. In TB itself, again, so simple modifications, and then you see such remarkable activities as reported. This compound, in fact, was one that I made as part of my PhD. But before then, the team had done quite extensive work. I mean, exploring all aspects of it, as you can see here. So they explored a lot. I mean, over 150 compounds in activity, in testing, to see what was happening. Now, here with this compound, we found that the compound had assay or media-specific activity, and we could rationalize that to say fusidic acid binds very strongly to the bovine albumin, which is part of the assay. And so we could rationalize why in albumin media we lost activity, but then in non-albumin media we could gain activity. So slight modifications, but then you are improving the potency of these compounds. And all these are based on facilities. If you look at the cascade, they are in one section of the cascade or the image I showed earlier, but all these are available to add value to what you do. Now let's talk about DMPK studies, still on fusidic acid. So the team, in fact, Matthew last week gave a talk on DMPK, and this work was part of his PhD work, in fact. So these are findings from Matthew, and he was able to find that in mice you would have fusidic acid epimerized to 3-epifusidic acid, which is inactive compared to 11 micromolar compared to 0.6 micromolar. However, in humans, you don't see this epimerization forming the opposite configuration, and the 3-ketofusidic acid is equally active. Okay, now the chemists made this compound, this silicate esters, and they were quite stable, although this particular compound loses some activity. The ethyl form of it was still very potent, comparable to fusidic acid itself, and these compounds were more stable. Okay, they did not undergo the epimerization as observed if you just have fusidic acid. Now some more work on PK and organ distribution. You see, so it tells you how, and this is spanning, if I'm not mistaken, over perhaps 7 years of research in the academic group. This was an active research, okay, making use of resources and facilities and infrastructure in H3D to pick a single compound, but then learn a lot of good information from it. Now, because of the poor metabolism of fusidic acid in mice, one can assume why in vivo efficacy studies has stalled for fusidic acid in spite of its very good in vitro activity. But the team used a prodrug approach where they made the acetal or the acetate form of it. And the idea here is that the acetate would easily enter and then release fusidic acid slowly so that it can prolong its activity. And this was proven. The team was able to prove it. But with time, again, you are going to have that the inactive epifusidic acid is going to still shoot up. But this is a good idea. It was a good plan using the prodrug approach. We have a good molecule. It has issues. Let's go back to the drawing board. What value can we add to it? Okay. So having done that, and this work speaks to some good work that my colleague Vinayak Singh did with his target ID expertise. Okay, so here he was able, at the time, we know that EFG or elongation factor G is the target in bacteria for fusidic acid, but we don't know that. I mean, someone has also reported that in malaria is the same target. But then through this research was able to confirm that in fact in MTB the same protein or the same target is hit by MTB, and was able to validate a new gene that was involved in this process. And in this figure, you would see that in his conditional knockdown experiment, you will see shifts in IC50 when it comes to fusidic acid. But then in isoniazid, you see that the IC50s, the curves are still the same. And so confirming that indeed this gene is implicated and then this target is confirmed. Again, speaking to the broad spectrum of work that one can do. Okay, I would end here and summarize this talk. I think that at this point you get the point, or you get what I'm trying to the message I'm trying to send across. Okay, when we talk of from nature to the lab to the clinic, the lab aspect of the journey is very important. We can benefit from nature, yes, but we need to make up our minds to have the lab component of it. Now you need the facilities, the resource, the infrastructure, the personnel, the know-how to be able to do that. And I want to present to the audience today, and for my colleagues here in Cape Town in South Africa and those across Africa, that we can do this research here in South Africa. Okay, and that H3D is a good place. We have it. So I put this, we are not called holistic for nothing. There is a reason why we are holistic. Okay, there is a reason why holistic is in our name, because we really do holistic drug discovery, which means that we have infrastructure, the personnel, the know-how. Okay, whether it is structural elucidation, we can help you. We are available to help. Whether it is mentoring, we are available to help. And we have the assays and all of that to help to improve or add value to what we have. Usually you have people go out and seek collaboration out of Africa, and they take the value out of us. But no, we can do it here. So from nature to the lab to the clinic is more likely to happen than from nature to the clinic. As I did show, just 4% from the period of 1981 till 2019 of compounds from nature have just ended up straight in the clinic. Even those had to go through the lab to be sure that they were the best. It is easier to find a potential natural product hit compound than to know how to maximize its value to make it a drug candidate. And this is where I would suggest we seek help. I can easily go on the internet and pick a compound I want to work with, or you can easily isolate a compound that you can work with. But it is how do you add value to it? Now that is where you need a team a team that has the know-how. And I end with this to say H3D is open to support, and I'm sure my team or my colleagues will be very happy to offer any support that is needed. So you are welcome to contact us. I would like to acknowledge the KC Academic Fusidic Acid team that did much a lot of the work that I've presented in this case study. And to you, Sruti, and the foundation, H3D Foundation, for the opportunity to remind ourselves of what lies ahead of us and what more we can do. Thank you.